Hello students, today's our topic in the chapter Principle of Inheritance. The topic is Pleotropy. What is the meaning of Pleotropy here? Pleotropy means when one gene responsible for more than one character. When one gene is responsible for more than one character is called Pleotropy here. And the gene which is responsible for the expression of more than one character is called pleiotropic gene. It's called pleiotropic gene here. To understand pleiotropy in detail, we just take a couple of examples here. First, sickle cell anemia. What's going on in sickle cell anemia here? Sickle cell anemia, it is due to hemoglobin. So if you firstly note it down that it is a recessive autosomal disorder. It is an recessive autosomal disorder. Here hemoglobin chain is made up of two alpha globular chains, means alpha globular chains, they are 2 in number, and 2 beta globular chains. Here either alpha globular chain affected or a beta globular chain affected, both are responsible for sickle cell anemia here. Next point is one chain affected in both cause sickle cell anemia. Here, so what's going on here? If we are going to talk about normal hemoglobin chain. So what's going on here? Here in this chain, there are some amino acid codes like valine, histamine, leucine, thionine, lectin, thioline, prolactin, glutamic acid, and we mark it as 1, 2, 3, 4, 5, 6, 7 and so on. Here 1, 2, 3, 4, 5, 6, 7. All base pairs are similar here. All amino acid groups are similar. Valine, histamine, lactane, thionine, prolactin, glutamic acid. But on the place of glutamic acid on the 6th position of amino acid. Remember, on a 6th position of a globular chain, there is a replacement by valine. Means we can say here, what do we have to say here? On a 6th position of hemoglobin molecule, on 6th position of hemoglobin molecule, or we can say globular chain of hemoglobin, on 6th position of hemoglobin molecule, glutamic acid is replaced by valine. So here glutamic acid replaced by valine on a 6th position is called sickle cell anemia. We just go through with the other causes of it. Next of all, here it is an example of point mutation. It is an example of point mutation here. Next one, if we are going through the causes of it, what are the changes occur? Changes. We have to write it here. Changes. First change. Functioning of hemoglobin molecule. Functioning of hemoglobin molecule. Second change is change the shape of RBC. Let us now remind on the first line. One gene responsible for more than one characters here. So two characters are, first is hemoglobin molecule functioning change and a change in the shape of RBCs, and a functioning means less attraction or a gaining of oxygen molecule. Carry less O2 molecules. Carry less O2 molecule here. And a shape, there is a normal, we firstly draw normal RBC shape, that is a normal RBC shape here. It's a normal shape of RBC, and when we go through with sickle cell anemia, wait, just correct it here, and one more. Sickle cell anemia shape. It is sickle like shape. It is biconcave shape. It is a biconcave shape where it is a sickle shaped structure here. So what are the probability For this, if we are going to denote hemoglobin S with affected and a hemoglobin A with normal, just note it here, hemoglobin S with affected, hemoglobin A with a normal here, then what is happening here? What's happening here? Firstly, we have three kind of sets here. First set is HbA, HbA, means we can say there is a homologous and a A representing here normal condition. So normal, homologous, normal. There is a no sickle cell in it. Second condition, HbA, HbS. What's now going here? In which due to HbA here, due to HbA, 50% RBCs normal. Okay, got it? And due to HbS here, 50% RBCs affected and having the structure like sickle cell, here. And the third and most dangerous condition, HbS. Remember here, it's a homozygous condition. Homozygous condition, homozygous affected. What's going on here? In a homozygous affected, it's a die, 100% affected here, 100% affected. Okay, here HbA, HbS, 50% affected, 50% normal, 50% sickle cell like. HbA, they are 100% normal. In this, it never will survive too longer, so it's a death here. So we are just going through with this, means in this condition only it survive for a longer time, and we can say it is affected with sickle cell disorder here, and it's a carrier because it is just to be look like normal, but it's having some kind of frictions like respiratory problem. It never we do a work in which high respiratory rate or high heart rate or heartbeat required for like jogging, for like playing outdoor games, for like running or jogging and hill climbing. He never be or she never be do the person with this such kind of characters never be do any kind of physical activity in which a high amount of oxygen required, because less affection of normal conditions of RBCs. That's why they are having a 50% sickle cell, and having 50% sickle cell, less capable to carry oxygen molecules here. So what we have to do in this? First of all, pleotropy. When one gene responsible for more than one characters here, they express more than one characters, so then that's why they are known as pleotropy, and such kind of genes which are responsible for such kind of characters are called pleiotropic gene. For doing the explanation of pleotropy, we just do an example of sickle cell anemia here. It's a recessive autosomal disorder, where recessive we know very well, which is never be expressed in the presence of any one of the dominant character, but autosomal. Here autosomal represent we having chromosomes like 23 pair. We are going to talk about pairs, means 22, first 44 plus XY in case of male, and a 44 plus XX in a case of female. The last pair of chromosome is called sex chromosome, and the rest of that, the above chromosomes like 44 chromosomes or first 22 pairs are called autosomes here. They require or they are responsible for many characters in humans. Hemoglobin chain is up to made up of two kind of globular chains. That is, first is alpha globular chain and another one is a beta globular chain. Both of these chains are 2 in numbers, means 2 alpha globular chains and 2 beta globular chains, and total number of chains in a hemoglobin molecule are 4, 2 alpha and 2 beta here. One chain affect in a both. If any one of chain affected in both cause sickle cell anemia here. Normal, if we are just going to go through with the normal condition of hemoglobin chain, then we just draw the normal condition of hemoglobin chain. In this condition, there are some amino acids. Amino acid 1 to 7. We just draw 1 to 7 amino acids here, in which on only 6th position of amino acid there is a normal condition of hemoglobin. 6th position, glutamic acid replaced by valine, and it's a caused or affected case of sickle cell anemia. Sickle cell anemia hemoglobin. So we just write down here on a 6th position of hemoglobin molecule, we just have to say on a 6th position of globular chain in a hemoglobin, glutamic acid is replaced by valine, and it is done only one place of the hemoglobin chain. So we can say it's a example of point mutation also. Changes first functioning of hemoglobin molecule and change the shape of RBC. What is the functioning change here? Functioning is the hemoglobin molecule carry 4 molecule of oxygen. Here in this case, hemoglobin molecule carry only 2 molecule of oxygen, not to be more than 2, or we can say that is not the function of a normal hemoglobin molecule. Second one, change in the shape of RBCs. There is a normal shape, there is a biconcave shape of RBC, and another one is a sickle cell anemia, sickle shape RBC here. What we have to do in this case, first of all, there is a hemoglobin molecule. These two are hemoglobin molecules. It's a normal condition as we have discussed earlier. There is a hemoglobin molecule. HbA is a normal condition of hemoglobin. There is a normal condition. Hb, HbS, in this HbA representing the normal conditions of 50% of RBCs are normal, where HbS representing the affected conditions of 50% RBCs are affected here. HbS hemoglobin molecule affected. HbS that is a hemoglobin molecule affected, so that's called 100% individuals affected with a sickle cell anemia, and such kind of person never will survive for a longer time. So we just assuming that the HbS means a heterozygous condition of a hemoglobin molecule causes sickle cell anemia because it carry 50% for the normal and 50% for affected. Now the next example of pleotropy is phenyl ketone urea. It is also a autosomal recessive disorder. It is also a autosomal recessive disorder. In this disorder, absence of an enzyme. Absence of an enzyme. The name of such kind of enzyme is phenyl alanine dehydroxylase. Means due to this disorder, a phenylalanine dehydroxylase enzyme is absent in the body. Now, what is the function of phenylalanine dehydroxylase? If we are going with, so it convert phenylalanine dehydroxylase, convert phenylalanine into triosine. So firstly we just write down a short reaction for this, and the reaction is phenylalanine. Here phenylalanine, which converted into triosine here, in the presence of an enzyme phenylalanine dehydroxylase. Means phenylalanine is a toxic substance which is formed in our body due to metabolic reactions, which is converted into triosine, which is less toxic in nature and go out through the body in a less toxic effect. So phenylalanine dehydroxylase convert phenylalanine into triosine here. If phenylalanine dehydroxylase is absent, then what will happen? If phenylalanine dehydroxylase absence, if phenylalanine dehydroxylase absence, then accumulation of phenylalanine occurs in the body. If phenylalanine accumulation occurs, then in such condition phenylalanine converted into phenylalanine converted into phenyl pyruvic acid. When phenylalanine accumulate in the body, then converted into phenyl pyruvic acid here. Or in such condition, we just talk about pleotropy, means one gene responsible for more characters here. There are no character we discussed. Now we discuss the characters. Here when phenyl pyruvic acid accumulate in a brain, it result accumulation in brain result mental retardation. here mental retardation occur. Second case, it reduce the pigment of hair. Reduce the pigment of hair. Next resultant, reduce the pigmentation of skin. Here reduce the pigmentation of skin also. So what we have to do in this? Next example of phenylalanine, the first one we discussed sickle cell anemia, and the another one is phenylketonuria. In this disorder, it is also a autosomal recessive disorder. In this, absence of an enzyme phenylalanine dehydroxylase. In such kind of enzyme which is responsible for the conversion of phenylalanine into tyrosine. Phenylalanine, we mark it as more toxic or tyrosine less toxic here. It converted phenylalanine into tyrosine, which is less toxic. If phenylalanine absent, as such we discussed in earlier first and a second point, that is it's a recessive disorder, and absence of phenylalanine dehydroxylase enzyme, it causes accumulation of phenylalanine in a body. If phenylalanine accumulation increases, phenylalanine in that case converted into phenylpyruvic acid. When phenylpyruvic acid is accumulated in brain, result mental retardation, or in some time it break down the capillaries and also cause brain hemorrhage. Reduce the pigmentation of hair and reduce the pigmentation of skin.